Hello, everyone, and thank you all for staying till this last session on this last day of JOBIM. I would also like to thank the organizers for giving me the opportunity to chair this session and to welcome my friend Rayan Chikhi to his first JOBIM talk ever, hopefully not the last. So I've met Rayan 12 years ago in Rennes, and at that time he was a brilliant PhD student finishing his thesis under the supervision of Dominique Lavenier. During his PhD, Rayan began working on sequencing data assembly, and maybe some of you remember Minier. That was a real breakthrough in the field. It was a frugal assembler Rayan developed with Guillaume Risque based on extremely smart idea that was using a Bloom filter enhanced by a hash table allowing to discard critical false positive chimers. And after his PhD, Rayan joined Paul Medvedev's lab as a postdoc. And in 2014, he joined the Bonsai team in Lille as a CNRS researcher. From 2019, Rayan leads a research group at the Pasteur Institute, which focuses on algorithms and data structures for processing DNA sequences. In the past years, Rayan's research has greatly evolved, and now he covers topics from genome to metagenome assembly to sequence search in extremely large data sets and also pangenomics. And so thank you for accepting our invitation, Rayan, and I leave you to it.
Merci pour l'introduction, Aloka. It's in English, right? So let's get started. Can everyone see the slides, right? I'm assuming that's a yes. And you also don't see the speaker, or is this some kind of a setup where you also see the speaker at the same time as the slides? Just so I know.
We can see both.
Oh, okay. All right. Let me see if on my setup. Okay. All right. Let's get started. The talk is called Living in the Future of Sequence Bioinformatics. It's going to be a relaxed talk. This is the end of JOBIM. Not too much technical. Thank you a lot for the invitation. I'm truly pleasured and honored to speak at JOBIM. So I hope you will like this talk, and let's get started. The introduction was already very nicely made by Raluca, so I won't say too much about it, except the pictures here are some pictures of organisms I contributed to assembling. This is giraffe, goat, gorilla, Y chromosome, axolotl, and so on. You can find me on Twitter, and this is my webpage address. So let's go right into the topic of the talk. Big data is, I claim, a natural flow, a natural continuation of biology. And the reason why I'm saying that is essentially a historical perspective. In 1972, the first gene sequence was sequenced and published. And if you see on the right, this is the paper that published this sequence. This is the P greatest P of P all P time abstract, because there is actually the sequence inside the abstract. That's the result. Nowadays, you cannot do that anymore because sequences are too long. And so the continuation of biology is that now there's more and more data. So in 2000, there was only one human genome, relatively high quality, that was published. And due to the explosion of short-read sequencing, 10 years later, many low-quality human genomes have been available. Fast forward to 2021, we analyzed 10 petabytes of data, of sequencing data that was publicly available. So that was another milestone of big data, I would say. Nowadays, there is in the order of millions of human genomes available in the form typically of VCF, so variant call formats. So not totally human genome sequences, but just some variant calls of SNPs and short indels. In terms of high quality human genomes, nowadays you have about 50 to 100 high quality, almost telomere to telomere quality. So you can see there's still a gap between the number of human genomes we have in terms of variant calls and the number of total genomes we have. And so data size is increasing. I guess the main question I want to ask is big data just a technical problem, or you can guess which side I am on, more of a scientific problem? I'm not the only one talking about this, and in fact, I was inspired by Richard JOBIM Keynote at Recomb Istanbul 2023, where he said something that really resonated to me. So of course, he said it better than me in terms of the big data perspective, but if you look at the bottom of the slides, he basically mentions that informatics is to biology what mathematics is to physics. And I think this really places our role as bioinformaticians or computer scientists in terms of what we can contribute to biology. It's perhaps the same way as mathematicians contribute to physics. You probably have seen or remember this XKCD comic that is a bit controversial, I suppose, amongst many fields, but I will just copy here to illustrate a point. So there is a scale of fields from sociology to mathematics. And if you look to the right, there is some sort of mathematical purity. Biology is somewhere in the middle. And if you look to the left, there is some sense of usefulness of the field. It's very provocative in both sense, saying that basically sociology would be the most useful field for society, which kind of makes sense. So in this comic, there is actually the three of the four fields I was talking about earlier: biology, physics, and mathematics. One might wonder where informatics and computer science is, and my take is that I would put it somewhere around here. It's highly personal, and you might totally disagree, but let's leave it to the question session. Coming back to the point, big data in biology, there are bigger and bigger databases nowadays. Some of them you probably already know very well. So GenBank is a repository of assemblies of 500,000 species and counting. It's now about 1 terabyte inside, and all the species are well annotated. That's a great thing. On the contrary, there's another database called WGS, also hosted at NCBI, which also hosts assemblies 10 times more than GenBank, but these ones are not annotated. Sorry, just technical setup change. All right, of course, there is the SRA database, which you probably know very well or have heard of. Nowadays, it's about 30 petabytes of public data. It contains reads, so raw sequencing data. All the reads that have been generated as part of a scientific study pretty much have to be uploaded to the SRA. So that's why the database is really growing and is an extremely useful resource. However, that's not the only one. There are also other databases in biology, such as UK Biobank and GTEx. And I was surprised by how much data Biobank actually contains. So like nowadays, it's about 500,000 human genomes. I would be curious how much, so this is really unclear if it can be interactive here, but I would be curious if someone has a guess of how large Biobank is in terms of the volume of raw sequencing data. So I guess JOBIM participation remotely is challenging. So I hear nothing or see nothing from the chat. Actually, I'm not sure if I can even really see the chat here. Oh, there's a chat. Yeah, there's nothing on the chat, so I'll just go ahead and give the answer. UK Biobank is now 25 petabytes in size, which is pretty much the same size as the SRA. So it's only humans, though not so much diversity, but lots of them. GTEx, however, which is a database of expression for human tissues, is a different size. And you probably can guess that it's, first of all, it has 10 times less samples, individuals, than UK Biobank, and also it's transcriptomes only, which is 10 times less data than a genome. So unsurprisingly, it's 100 times less than Biobank and is about 100 terabytes. Still big data by many metrics. So all of these metrics with tera, peta, and so on can be a bit confusing, mind-boggling. Here are the explanations, and I'm drawing the line at exa because that's pretty much the volume of data that nowadays we can handle. Of course, we as humanity, right? So nobody has an exabyte of storage. But the point here is that we cannot really go farther, and nobody talks about zettabyte just yet, but maybe we will in a decade or so. To give a sense of scale, your laptop has 0.001 petabytes of storage, which is 1 terabyte typically. Institut Pasteur has a few tens of petabytes now. The image is a bit dated, but it's still smaller than the NCBI SRA database. Of course, the NCBI building is larger, so it can host more data than the Pasteur building. If you compare to the size of data in other fields, such as video storage, as YouTube, biology is still catching up. YouTube is still an order of magnitude larger, but this might change in the future. The SRA is not stopping in terms of its growth. So far, it has doubled every 2 years. So nowadays, it's 30 petabytes in 2024. At the end of it, it will be 60 petabytes. The clock is ticking, and the size keeps growing. As a reference to the keynote from Anaïs yesterday, when she was asking whether some of those data was big data, well, I suppose for what I presented earlier, the answer is clearly yes. This is really too big data to be analyzed for biobank, SRA, and so on. The claim of this talk is that with big data and big computers, one could perform wonderful, groundbreaking genomics. I'm trying to inject some positivity at the end of JOBIM, so please focus on this arrogant yellow balloon and think of the wonderful things you could do with big data and big computers. However, the talk is not finished yet, and I'll try to answer a little bit how this kind of analysis could be done. And for this, I really like this quote from the creator of Gmail, who says, so he's not a bioinformatician by any means, but I think it applies. People at the leading edge of a rapidly changing field live in the future. So what does that mean? Consider another quote from a philosopher this time. If you want to know how to paint a perfect painting, it's relatively easy. Just make yourself perfect and then paint naturally. Don't take this quote too literally, of course, but the point here is perhaps the same as the previous quotes. So it's about really living in the future and changing your perspective on things, and this will lead to a different sort of science being made by the person. I will spare you some of the quotes. I think Pasteur has also made some quotes in the same spirit, but I will stop here with the philosophy and let's actually get started on the concrete, what it means. What does it mean to live in the future in biology? It means a few things. So perhaps you might have a lab technique that is not very widespread. You might have data that only you have. You might have a theory or a belief that is not established yet. So in a sense, you live in the future. Or you could work on sci-fi projects such as creating a cell from scratch. And I'm sure there are other ways to quote unquote live in the future. I would be happy to hear about it in the comment section of this video on YouTube, I suppose, or on the question session of JOBIM. So that's biology. What does it mean to live in the future in bioinformatics and computational biology? Well, it's similar actually. So you might have a computational technique that is not very widespread. You might have data also that's not public. And you could also work on sci-fi projects such as analyzing data so big that no one would believe it can be done. Let's have a look at how some people are living in the future, in my sense. So if you consider people like George Church and Craig Venter, they're trying to create cells. That's just plain science fiction. Karen Miga and the T2T consortium have been finishing the human genome. So for a while everyone else, myself included, was using old school human reference genome like GRCH38 or HG19, which was okay, but still had gaps, while Karen Miga and colleagues, they were using a perfect human genome reference with no gaps, complete centromeres. So they were definitely living in the future. And arguably they still are, because not everyone has transitioned to CHM13 as a human reference genome. However, they should. Evan Eichler and Eric Garrison, with their pangenome work on considering variants across populations of human genome, in a sense, they are living in the future. But my overall point is that every one of you in this room, all researchers, we are all living in the future in a sense, because we are months ahead of the rest of the community with our results, with unreleased papers and some of the theories that are not yet published. But more concretely, so future genomics, what would it look like? I claim that one way to achieve a future type of analysis is to use future computers. And for that, I will give you a little demo if time permits, and I suppose it will. So the claim here is that in data centers, the number of CPUs has steadily grown across the years. Even if CPUs are not very faster per core, the number of core is greater. So 6 years ago, we had only 32 cores per server. Now we have 128. So that's really great. And what can we do with it? Let's consider some example. And the simplest example I could think of is getting reads. So in the past, if you wanted to get your sequencing data, you could only use a Pigeon Voyageur, and that was extremely inefficient. So how do we get fast queues nowadays? Well, we download reads to a laptop or to a cluster. And let's give a small demonstration here. So there are these reads. They come from a human sample. They are sequencing using PacBio i5. This is the URL of those reads. They are on S3, so that's an Amazon repository. I wanted to download these reads. I would just go to my download folder and type a command. Actually, hope you can see it well. And I copy paste the URL. I say I want to do it here. And of course, I messed up with S3CP. When you download those reads, you grab a coffee and you wait a few hours or maybe a few tens of minutes, because it's only downloading at 9 megabytes per second. So I will not wait until the end of the download, but you can imagine that it's a bit painful. Let's just stop here to download. There's actually a better way to do it. And the way I claim is a bit futuristic way is you don't download the reads. You use only local reads, the ones which have been created and homegrown in their own country. So you don't make the reads leave the country, you go to them. And how do you do that? By renting computers which live in the same location as where the reads are. So consider a computer right here, which actually, let me just connect to it to show. I SSH a certain computer with an IP address in the US. It's one in Amazon. Of course, it runs Amazon Linux because I like it. And now if I do the same copy as before, you will notice that it goes way faster. No longer 9 megabytes per minute, but instead close to 300 megabytes per second. Sorry, 9 megabytes per second versus 300. So that's more than an order of magnitude faster. And now we can finally get those reads within the time it will take to do this talk. While this is downloading, I'll keep going. So the point here is that it's highly useful to use the cloud. What's the cloud? It's a collection of computers owned by a single organization, accessible from the internet. So of course, I love this picture on the right, but I also love the picture on the left because it's one of the only pictures of a cloud you can see. Amazon doesn't let you take pictures, but OVH in Roubaix, no problem. Actually, I see a chat message, just a second. Is Q&A? I guess it's not Q&A. Le bridage est-il la seule raison du business model d'Amazon? I guess the Q&A is for later, but I will still answer it now. No, it's not a bridage here. It's really because my connection in this hotel room where I am in Penn State is not good. And even if you try to download to a cluster in France, it will have to cross the Atlantic if the reads are from the UK or from the US or across the Manche. And that's typically not fast. The only way why this is fast is because actually the reads don't really escape the data center. They are coming from SRA pub SRC2, which is a bucket in the same data center where I rented the machine. And typically, when you rent cloud machines, you can select the power of those machines. So if you rent a T3A.large, which is a bit of an obscure name at Amazon Web Services, you get 2 cores and 8 gigabytes of memory, which is about your laptop. So there is this naming convention that you can spend hours staring at, but generally the number after the dot is indicative of the power. So you can see that the 8x large is twice as powerful as a 4x large, and so on. A machine I really love is C6A.40x large. It's a beautiful machine. It has 192 cores and about 400 gigs of memory. Really, I think it's one of the most powerful, if not the most powerful one at Amazon. And that's actually the one that's running right now. So these have been downloaded. That was relatively quick. Let's get going with the demonstration. A little parenthesis. So what is nearly a cloud, but not really a cloud? So your university cluster has almost all of the cloud capabilities. It has many computers. They often sometimes let you be root on some of those computers if they have some OpenStack or something set up. If you're not root, you're not really using a cloud. This is a key point. If you have 7 Raspberry Pis stacked together, you have a cloud, which is not very powerful, but it's technically one. And of course, this dog is almost a cloud. What does it mean to store information in the cloud? It means that the data resides on the disk of a computer somewhere connected to the internet. And typically, there are plenty of computers that can access this disk very fast. So there's nothing mysterious about this sentence, but it's important to realize it. Concretely, if you look at US clouds, for instance, there are 2 big data centers. One is called US East 1, the other one is US West 1. And the key thing is that the SRA data at NCBI is stored very close to US East 1 geographically, and also has been mirrored physically inside US East 1. However, the T2T data from the Human Pangenome Project and also CHM13 is not in the East Coast, it's in the West Coast. So if you consider that maybe there's some distance between those data centers, it may not be a good idea to use an East Coast data center to download West Coast data. Does it make a difference? Well, I did some tests, and the ping time between those 2 data centers, which is indicative of the speed of light, essentially, is about 70 milliseconds. And you know that a hard drive can read or write at about 2 gigabytes per second. And when I did some tests, when you download data from the same data center, it's about 400 megabytes per second. I don't know why today it was a bit slower. When you download between 2 regions of the US, it's about 200 megabytes per second. So it doesn't make that much of a difference, actually, as long as you are in the same country. Let's see if we can actually do this. So there's a small fun exercise for the audience. Try to connect the dots between the different tasks on the left and the different times it takes to do those tasks on the right. What I propose is I give you a few seconds or a minute, and whenever somebody shouts the answer either to their microphone or to the chat, I will continue. Already, 21354. Okay. 354. Yeah, that's the correct answer. So let's get there. So access data in memory is very fast. It's the fastest thing you can do, about a fraction of a microsecond. significantly longer it takes to read stuff from a disk because of random access to disks, so it's in the order of milliseconds. And also significantly longer is accessing data from the web, especially when it's around the globe. So 200 milliseconds is the ping time, more or less. Aligning 1 million short reads is, of course, requires to do pretty much both of the first two tasks, so many times. So it takes 10 seconds with, I think it was BWMM. And human cell cycle, just to say that biology is really slower in general. So it takes 24 hours for a cell to duplicate. The point of all of this is about speeds. And the question I want to ask now is, if we have a lot of CPUs, for instance, 200 CPUs in this machine, can we go 200 times faster than if we had a single CPU? At being faster at general bioinformatics tasks to make the question more precise. And regardless, the answer is typically no. So there is a law of somebody called Amdahl that essentially states when your efficiency, sorry, it's in Spanish, so it is the international JOBIM now. If your efficiency at being parallel for a certain task is 95%, so that means you can pretty much do treatment in parallel except for 5% of your software, then your acceleration is capped. It doesn't matter how many CPUs you're using, you will only be able to, actually, do you see my cursor? I think you see it here. You will only be able to go as 20 times as fast as if you had only one CPU. And 95% is pretty good, so sometimes you're only 50% efficient at being parallel, and then it means that you cap your acceleration at 2. So essentially, this effect makes you have diminishing returns at how many machines you throw at a problem, or how many CPUs you throw at a problem. Let's consider a concrete task. The task is to map some reads to the human genome. So imagine you've sequenced yourself using PacBio Hi-Fi. You want to do some genetic tests on yourself. You are a bit in a hurry. You are in the future. How long would it take? Unfortunately, we are not yet in the future. And today, with Minimap2, if you have a machine with 10 threads, it takes about 1 hour. Is this really the future we want to live in? I would say no, this is not. 1 hour is too slow. We want to do this basic task very faster. How can we do it faster? Well, using maybe future computers. So let's try this. So now we have Minimap2, and let's try to map it with Hi-Fi setting. Actually, let's not go with -A, let's go with this. And I have prepared a human reference genome here. This is a P2P reference. And this is the read file that I just downloaded a few minutes ago. The key point is I'm going to use 192 threads and store the result in a file. So that's Minimap2. We can observe how long it's taking with top. So right now it's using only one CPU because Minimap2 has a step where it's indexing the human genome. These steps typically take a few tenths of seconds. Or more, I guess. Okay, now it's collecting minimizers from the human genome, it's creating an index, and that step is not really parallel. And now it has built an index, and now it's actually starting to map the reads about now. And when it does that, you can see that it's really actually speeding up significantly. And a good thing to watch is actually how the CPUs are being used. So this machine has 192 CPUs, and each top shows you the CPU usage of all of them, a fraction of them. And now this is a machine put to good use because it's really not using 100%. There is not a 100% efficiency at parallelism in Minimap2, but it's fairly good, except right now, but it's fairly good in general. So now it's really using those 192 CPUs as evidenced by top. So if I kill this, Then you can ask yourself, so how much time is it going to take to map this 10x human genome, or 10x coverage human genome reads? Well, if you had a normal machine, it would take an hour. But you can guess that since I'm running it now, it probably will not take an hour. Otherwise, the keynote would be extremely boring from now on. If this is a rhetorical question, you don't need to answer it in the chat. Really good. All right, so it took 2 minutes. That was a satisfactory experience. So that's part 2 of the demo, Minimap 2 on steroids. The key part is that you use a really big machine to do mapping of hi-fi reads, and you get an order of magnitude speed up because your machine is an order of magnitude faster. So I use the T2T genome, I pre-downloaded it, I saved you the pain of downloading it, which is available here, also on AWS S3, and it's very quick to download. Small mistake here, it's not a SAM file, it's a path file. Path means that you actually don't store any alignment information, you just store the coordinates of the reads to the human genome. All right, so that was hopefully satisfactory, but you might say, okay, I don't have 2 minutes. Can I do it even faster? Answer is yes, you can actually use a software that I co-developed with Barry Shekim called MapQuick that is really specific to mapping hi-fi reads. Let's give it a try. So this is MapQuick. If I remember do I remember actually the command line? I hope so. This is THM13. Then I give it the reads. And I want to use, actually, I'm going to be generous here and give only 10 threads because we are in the future, we have better algorithms, we don't have better machine. Let's suppose this. Map it to prefix. So it's starting, it's indexing the human genome. It's faster than Minimap 2, that's self-promotion right here. And now it's doing its alignment, its mapping task to the human genome. Small thing to note, well, not actually, no, I will not go into the algorithmic details of what's going on right now. Because that would be for another talk, so invite me again if you would like to hear about it. So it has finished in 30 seconds, 10 threads, and it has aligned 10x depth of human hi-fi reads to the human genome. I hope that was satisfactory to you as it is to me. Okay, so that's part 3 of mapping quick to the human genome. I will just briefly, before I wrap up this talk, mention another kind of future project that's close to my heart, which is unrelated to alignment this time, but it's about searching all of life sequencing data, so all of SRA, through the creation of an index. So if we had an index of all of SRA, all the samples of SRA, it could be the raw data or assemblies, and you could search it, then you could perform queries of sequences, such as saying, does this sequence occur in some of the datasets of SRA, and if so, which ones? And the way to do it typically is using K-meres, so you search for a certain subsequence. The index tells you where that subsequence is within some sort of dataset. So this index doesn't exist. I want to create it. This is the purpose of an ERC project that will recently start. And it's essentially the same thing as Google, as it does for web pages, but here we would create a Google for the SRA. It's not really an original project. Many people want to do it, but the problem is mainly scale. So it requires big machine and also big algorithms. All right, so the outro is pretty long, so I have a dedicated section for it. Bear with me. So now we are escaping the world of big data, and I'm claiming that we are going into the bigger data territory because it's even bigger than before. For this, we are going to need even bigger instance types than the one I just demonstrated today. Oh yeah, this talk, I already gave this talk once to the workshop of genomics, so I dedicated to these people, but I also want to now dedicate, secondly, this talk to JOBIM. Thanks a lot for the keynote invitation. I couldn't find a 2023 photo, so I took a 2012 photo. Bear with me on this one. And so this is my group at Institut Pasteur. If you're interested in this kind of analysis, feel free to reach out. We do several things. The mascot of the group is an accelerator. We work on assembly, data structures, large sequence search, which was the purpose of this talk, and in a way, pangenomics. And I thank you a lot for your attention. Happy to take any questions.
So thank you, Ryan. I hope you hear me.
Yeah.
Thank you for yet another excellent, quite original presentation. So, I don't know whether there are questions. Okay, so I have a question on the chat. What do you think of the future contribution of quantum computers in bioinformatics?
Is this a chat question? Okay, I don't see it.
Yeah.
So, quantum computers, yeah, they are. I get these questions sometimes. It's a bit science fiction. There are some theoretical works of people investigating quantum algorithms in bioinformatics. None of them have had any practical impact yet. Maybe one day they will. Until we actually see a quantum computer performing a general purpose task, which is not physics related, we are still in the domain of science fiction. Nowadays, with quantum computers, they mostly do physics simulations, and they are good at this. But that's not totally useful, or especially that's not useful at all for bioinformatics. So for now, let's try to do some nice research on classical computers. And when we get there, we will use quantum computers. One thing to note is that the theory of computing in terms of quantum is already very well established, very well known. The quantum complexity of algorithms has been studied for several decades. So people have been exploring it. And there are limitations, of course, and also potential opportunities. But I think it would be foolish to stop funding classical computing now in the hope that quantum computing will come, because this hope is very distant.
Okay, there is another question regarding the cost per hour of the machine you showed at the Amazon.
Yeah, absolutely. So is my screen still sharing? I think it is.
Yeah.
Okay, so let's have a look. This might be too small. So there's a web page you can access if you type a few hidden keywords on Google, which is AWS and Vantage. That's the name of the website. It gives you the cost and specifications of all of the machines you can rent on Amazon. So the small ones you can see, they cost can I zoom in here? They cost $0.002 hourly. That's on demand. It's typically on demand. You want to do it. And if you go for the biggest machines, you just sort in that. Those 400 CPUs, you can't really rent them. They are metal thing, and they also are prohibitively expensive, so I will never give it a try. Let's see, the one I rented was C6A40X large. That's 192 CPUs, and it cost $7 per hour on demand, and a bit less if you do some reservation, and a bit less if you allow it to stop at any point. So that's the order of magnitude of cost.
So are there any questions from the different sites, from Nice, for example? While you're trying to get some questions, I would have one general question. And I guess regarding your future projection, I can imagine some of the technical and societal obstacles that you have to overcome to get to all that you imagine in terms of using this data. But could you just develop a little bit on the remaining scientific challenges? I mean, from the point of view of the data structures, algorithmics, the locks that are still remaining.
Yeah, no, that's a great question, and in fact, you're asking for the secret sauce of the project. So I can say a few things about it. I don't want to be too private about it, but there's this problem of indexing a lot of keywords, which has absorbed the bioinformatics community and many French researchers for at least a decade. And we have kind of hit a wall in a sense that using any computing architecture that we have, excluding quantum computers, but again, they don't work, we cannot index a large amount of keywords. By large, I mean more than a few petabytes of raw data. And you know that SRA has more than a few petabytes. It has tens of petabytes. So the best methods that we have right now are totally limited and cannot process the SRA. So there is my strategy in this project would be to break it into small steps. And the first step would be to start by assembling each of those accessions one by one. And you know that when you assemble an accession, you greatly make the data size smaller because you don't have reads anymore. You have consensus assemblies or even conservative assemblies, which are unitics. They don't lose any informations in terms of keywords, but any keyword that is repeated in the data set is only seen once in a conservative assembly. Whereas in a raw data, it is seen 30 times, 100 times, even more. So let's start by the data reduction step. But I would say that even this data reduction step is not trivial because processing tens of petabytes of data, well, it requires advanced architecture and also some smart algorithms. We've done this in a Serverless project for alignment, and here I want to do it for large-scale assembly. Once we have these assemblies, the challenge of creating an index is another one, but I don't want to be too long on the answer and stop here for now. But thanks for the question.
Thank you for the answer. And so, do you have some questions from Nice, from Tapitre?
If nobody does, it's 10:10 now. It's like 4:10 in France, so I'm on time.
I still have one question. If
Okay.
we still have some time. Okay, so for your ERC project, congratulations, in fact, for it. So
Merci.
you mean indexing and querying all this ENA SRA data. And I guess here you mostly talk about raw data, so raw sequencing that I could also call flat sequence data. I don't know if it's the good term, but maybe you'll get it when I get to the end of the question. So what I mean is, what about more complex types of sequence data, like the ones we have now in pangenomics? So pangenomics reference, for example, do you mean querying this kind of data also? And
yeah, that's a good point. So, well, if you think about it, the pangenome data is a collection of samples from the same species or same group of a group of individuals of interest. In a way, you can think of SRA as a very large collection of pangenomes for all of life. So, like for instance, if you take the subset of the SRA corresponding to isolate microbial data set for E. coli, that's an E. coli pangenome, except that it is not really assembled, it's in raw data form, so you don't have the complete genomes. But for many biological applications, it's not necessary to have a nice pangenome graph. You can also just want to query some variations inside the reads, and the variation is still accessible as certain keywords, for instance. So I guess it's not totally incompatible what you're asking about, so querying pangenomes and querying the SRA. In a way, you could ask also, what can an index like this be created for the UK Biobank, which is the biggest human pangenome we have nowadays? And there's one technical challenge here is that the UK Biobank is controlled access. So yeah, it's not public, it's sensitive information. So making such an index accessible would bring another set of ethics challenges that I don't want to tackle for now. So I only want to deal with data which has given its consent to be accessed publicly, and typically bacteria tend to give consent very easily. So human pangenomics are not going to be a part of this project for now for the problems of essentially confidentiality of the data.
Okay, thank you.
Yeah.
Do we have questions?
There is a question.
I don't see. Oh, sorry. Okay.
Sorry. Yes, there is a question from Pointe-à-Pitre. Very nice talk. Thank you, Rayan.
Thank you.
Yes, I would like to know if there is a difference between indexing. What are the differences between indexing SRA data as compared to assemblies?
Yeah, good point. So indexing assemblies is as old as bioinformatics because BLAST typically is an index of the RefSec assemblies. Well, it's not quite RefSec, it's a clustered version of RefSec. So they took all the assemblies that have been generated and annotated so far, they remove redundancy, and they create an index. That's essentially what BLAST is. It works very well. It's somewhat limited because you cannot, for instance, as Raluca said, you cannot enrich the index with pangenome variation because you are limited to one exemplar per species typically. So to answer your question shortly, it's a question of scale. So indexing SRA is many, many orders of magnitude larger task than indexing what we have currently assembled as good quality genomes.
Okay, thank you. Thank you.
Oh, I see a question from Matthias.
Do you see the question, or do I need to read it maybe for the audience? So
sure, I can read it.
Okay.
He said, what kind of SF question? Well, first of all, he said wonderful talks. Thank you, Matthias. Appreciate it from you. What kind of SF question do you think you could solve using SRA querying? Yeah, essentially the question is why do we do this? Huh? Is it just fun, or are there some applications? I would argue that there are some applications. So a subset of the SRA has already been indexed very recently by Pierre Petrolongo and Theo Leman in a software called KM Index, which I am a co-author of, so this is self-promotion as well. And this subset is Tara Ocean data. And it seems to be quite interesting to index it to perform search for genes inside these Tara samples. So hopefully an index of Tara sequences will give some insights on oceanography. But SRA is much bigger than Tara. So the sort of things we could do, we've already done, in fact, with SRA investigations, is discover new viruses. So we did that a couple of years ago in the Ceratus project. We essentially queried the SRA for any sequence that looks like a virus, and we discarded all the sequences which were exactly like the virus we already know, and instead we kept those that had some distant homology to viruses. And when we assembled those sequences, we could realize that there were, in fact, many, many viruses hidden inside the SRA that people had sequenced incidentally, but that wasn't the main point of their sequencing. So basically, when you query the SRA, when you reanalyze all the data that has been produced by humanity so far, you can do discoveries and find new species of viruses, but not only, you can also discover new bacterial species for sure. and especially in oceans and in soil. And the only way you can do this is really to harness the power of all the data that has been created so far, because if you only analyze a small set of data, you will discover less. I hope it answers part of the question. There are other applications, but I don't want to be too long. Okay, I see a thumbs up, so that's probably a yes.
Okay. I don't know whether there are more questions. If not, we can indeed stop here just to gain some time on the next of the conference. So thank you once again, Rayan.
Thank you. And goodbye, JOBIM.
Have a nice trip in the United States.
Yeah, I hope so. It's early morning here, so it's convenient to talk at JOBIM remotely, since all of you is remote anyway. Okay, I'll give back the screen and stop chatting. Thanks a lot, everyone.
Thank you.
Merci beaucoup, Rayan. Merci, Raluca.